Diseases of lens. Today let's see the introduction part. Anatomy and physiology. Applied anatomy. Shape, transparency and location. Lens is a transparent biconvex crystalline structure placed between iris and vitreous in a saucer-shaped depression. The patellar fossa. Diameter is 9 to 10 mm. Thickness varies with age from 3 to 5 mm at birth to 5 mm at extreme of age. Weight varies from 135 mg 0 to 9 years to 255 mg 40 to 80 years of age. Surfaces. Anterior surface is less convex. Radius of curvature is 10 mm than the posterior. Radius of curvature is 66 mm. Refractive index is 1.39. Total dioptric power is about 18D and the cornea constitutes about 40D. Accommodated power of lens varies with age, being 14 to 16D at birth, 7 to 8D at 25 years of age, 1 to 2D at 50 years of age. Lens capsule. It's a thin, transparent hyaline membrane surrounding the lens, which is thicker over anterior than posterior surface. The lens capsule is thickest at pre-equator region, 14 micron, and thinnest at posterior pole, 3 micron. Anterior epithelium. Let's see the structure. This is nucleus. It develops slowly from embryonic to adult stage. And this is cortex. This is anterior epithelium, anterior capsule of lens. Anterior epithelium, it is a single layer of cuboidal cells which lies deep to anterior capsule. In the equatorial region, these cells become columnar, are actively dividing and elongating to form new lens fibers throughout the life. There is no posterior epithelium as these cells are used up in filling the central cavity of lens vesicle during development of lens. Okay. Let's see about lens fibers. The epithelial cells elongate to form lens fibers which have a complicated structural form. Mature lens fibers are cells which have lost their nuclei. As the lens fibers are formed throughout the life, these are arranged compactly as nucleus and cortex of the lens. Y-shaped sutures of fetal nuclear fibers. Then nucleus, it is the central part containing the oldest fibers. It consists of different zones which are laid down successfully as the development proceeds. In the beam of slit lamp, these are seen as zones of discontinuity. It is important. Depending upon the period of development, the different zones of lens nuclei include embryonic nucleus. It is the innermost part of nuclei which correspond to lens up to first 3 months of gestation. It consists of primary lens fibers which are formed by elongation of cells of posterior walls of lens vesicle. Fetal nuclei, it lies around the embryonic nucleus, corresponds to lens from 3 months of gestation till birth. Its fibers meet around sutures which are anteriorly Y-shaped and posteriorly inverted Y-shaped. Infantile nuclei correspond to the lens from birth to puberty. Adult nuclei correspond to lens fibers formed after puberty to rest of life. Cortex, it is the peripheral part which comprises the youngest lens fibers. Suspensory ligaments of lens. Suspensory ligaments of lens, zonules of Ginn, also called as ciliary zonules, consist essentially of a series of fibers passing from ciliary body to lens. These hold the lens in position and enable the ciliary muscle to act on it. Arrangement of zonular fibers. According to new concept, zonular fibers run a more or less complex but continuous course from ora serrated to edge of lens. However, the purpose of description only, zonular complex can be divided into following parts. Pars orbicularis refers to feltwork of zonular fibers which line pars plana, part of ciliary body. Zonular plexus refers to intervening network of zonular fibers which are attached between ciliary process in the region of pars placata, part of ciliary body. Zonular fork refers to consolidated bundle of zonular fibers which bend at right angle from the anterior margin of pars placata towards the lens after dividing into 3 zonular limbs: anterior zonular limb, equatorial zonular limb, posterior zonular limb. Anterior is denser and insert on anterior lens capsule about 1.5 mm from the equator. Equatorial zonular limb are sparse and fan out in a brush-like manner to get inserted into lens capsule of equatorial region. Posterior jonular limb fibers are inserted in posterior lens capsule about 1.25 mm from the equator. One is pars orbicularis, two is jonular plexus, three is jonular fork, 4 is anterior jonular limb, 5 is equatorial jonular limb, 6 is posterior jonular limb. Then applied physiology and biochemistry. Lens transparency, metabolic activities of lens, and accommodation. Lens transparency. Factors that play a significant role in maintaining outstanding clarity and transparency of crystalline lens are avascularity, characteristic of lens fiber, tightly packed narrow lens, loss of Arnold's, role of lens proteins. Major interesting proteins of lens belong to superfamily aquaporins and is classified as aquaporin MIP26 or aquaporin 0. Lens capsules, semi-permeable character, pump mechanism of lens fiber membranes that regulate the electrolyte and water balance in the lens, maintaining relative dehydration. Auto-oxidation and high concentration of reduced glutathione in the lens maintain lens protein in a reduced state and ensures the integrity of cell membrane pump. Metabolism. Lens requires a continuous supply of energy, ATP, for active transport of ions and amino acids, maintenance of lens dehydration, and for a continuous protein GSS synthesis. Most of the energy produced is utilized epithelium, which is major site of all active transport process. Only about 10 to 20% of ATP generated is used for protein synthesis. Source of nutrient supply. Crystalline lens, being AFR structure, is dependent on mechanism, metabolism, and chemical exchange with aqueous humor. The chemical composition of lens with aqueous humor and chemical exchange between two is depicted. Let's see further. Then some nutrition is also derived from vitreous humor, but it is much less than that derived from the aqueous humor. Pathways of glucose metabolism. Glucose is very essential for normal working of lens. Metabolic activity of lens is largely limited to epithelium and context, while the nucleus is relatively inert. Glycotic pathway. In the lens, 80% of glucose is metabolized anaerobically by this pathway. Exhaust monophosphate pathway, HMP shunt, responsible for metabolism 15% glucose. Krebs cycle metabolism only a small proportion of oxidative process. Then let's see chemical combustion of lens vis-a-vis aqueous humor. Chemical exchange. What happens? Water, sodium, potassium, chlorine, glucose, lactic acid, glutathione, inositol, amino acid, proteins all are exchanged. These are the concentrations. Orbital pathway. It is relatively inconsequential in normal lens. However, it is extremely important in the production of cataract in diabetic. Antioxidant mechanism of lens. Crystalline lens has protective mechanism against oxidative damage caused by various oxidants. Oxygen intermediates generated in vivo, such as superoxide anion, hydrogen peroxide, lipid peroxide, lipid hydroperoxide. Of these, superoxide radicals may be most toxic to lens protein. Preventive antioxidant mechanism of lens. Enzymatic defense mechanism, which include reduced glutathione peroxide, superoxide dismutase catalase. Non-enzymatic defense mechanism include vitamin C, vitamin E, carotenoids. Pathogens of age-related cataract involve oxidation of lens protein and lipids due to deficient preventive antioxidant mechanism. Let's see about cataract in part 2 of the video.